Assalamu alaikum. Uh, I'll be teaching you today a topic from Biology O-Level syllabus 5090. And before that, I would like you to introduce myself. My name is Dr. Tanveer Qazi. I am a medical doctor as well as a teacher by profession. I did my MBBS from Khyber Medical College. And then after that, in the National Institute of Child Health, JPMC Karachi, I joined for my post-graduation and passed my diploma in child health examination from Karachi University. Later on, I joined the Army Public School, Sadr, which is an O-level school, as a school doctor, and they also offered me to become the subject head for biology. And I started teaching biology and kept teaching for the next 5 years. Later on, I joined the City School PF chapter as their subject head for biology O-levels and remained so for 10 years. And so I have a lot of experience of teaching biology O-levels and have many of my students have become high achievers and they pursued their careers successfully later on also. I was later on the school doctor at Huck Academy for 7 years, but I kept teaching biology to O-level students privately. Currently also I'm teaching privately and also in reputable institutes of Karachi. So our topic for today, which I'm going to teach you, is about photosynthesis. Photosynthesis, you know, is one of the reasons because of which life exists on Earth today. It is responsible for life of plants and living beings, all living beings that are there. Now let us define photosynthesis. It is a process in which light energy is absorbed by chlorophyll and transformed into chemical energy, which is used in the synthesis of carbohydrates from water and carbon dioxide. In this process, oxygen is liberated. Water and carbon dioxide are also called the raw materials for photosynthesis. But from this definition, you can find out what are the requirements for photosynthesis. First of all, sunlight or light energy. Then we need chlorophyll, which is the green pigment in the plants. We also need carbon dioxide from the air, and water is absorbed from the soil, as well as suitable temperature is required. Why is suitable temperature required? Because there are a lot of enzyme reactions taking place inside the leaf, and you know that enzymes need a suitable temperature to function. Now, there are two stages of photosynthesis. The first one takes place in the presence of light energy, so it is called light-dependent stage. Light energy is absorbed by chlorophyll and converted to chemical energy. Also in the light-dependent stage, light energy is required to split water molecules which have been absorbed from the soil into its components, which are oxygen and hydrogen. This splitting up of water molecule into its components is called photolysis. Now, the next stage of photosynthesis can take place in the absence of light. So we also call it light independent stage. Now, as we know, we got splitting of water and we got hydrogen molecules. Now, the hydrogen which is produced, the atoms which are produced from the first stage are used in the light independent stage to reduce carbon dioxide to carbohydrate or glucose molecules. For this process, the energy which is provided also comes from the light stage or the light dependent stage. Now, in your syllabus, you are asked to write down the equation for photosynthesis. Now, you can either write down in words, carbon dioxide plus water in the presence of sunlight and chlorophyll is converted to carbohydrate or glucose and oxygen. Or you can write a chemical equation, 6 molecules of carbon dioxide combined with 6 of water in the presence of sunlight and chlorophyll. We get 1 molecule of glucose, C6H12O6, plus 6 of oxygen. This is the oxygen which is liberated in the air. Here is a diagrammatic representation of the process of photosynthesis. This is a green plant. Green plants have leaves, green leaves, which have chlorophyll present in the chloroplast. Now, carbon dioxide is taken from the air, sunlight is being provided to the plant, and water comes from the soil. And in the light and stage, we get photolysis, and in the light independent stage, we get the molecule of glucose. Oxygen is being liberated during the process. Now, in your syllabus for photosynthesis chapter, we have got a number of experiments. The first series of experiments needs you to show that what are the essential requirements and how can we demonstrate that sunlight is essential for photosynthesis, chlorophyll is essential, and carbon dioxide is essential for photosynthesis. But first, before performing these experiments, you need to know the test for starch in a leaf. Why do we do that test? Because if photosynthesis takes place, sugars are formed. Now, these are used by cellular respiration. The glucose is used for cellular respiration by the plant itself, but the excess glucose is converted to starch and is stored inside the leaves and other parts of the plants. Now, when this starch is stored, we can test it. But before doing the experiment, we must de-starch the potted plant. And how do we do it? We place, to de-starch a potted plant, you place it in darkness for nearly 2 days. Now, how do we test for starch in a leaf? We take a green leaf from a plant that has been exposed to sunlight for a few hours at least. Put this leaf in boiling water for 2 minutes. This can be in the beaker. This process kills the enzymes which convert starch to sugar because we don't want the starch to be hydrolyzed. We put the leaf in boiling tube, take out with the forceps and put this leaf in a boiling tube containing alcohol. You place it in the beaker of hot water, but the precaution taken here is you must turn off the Bunsen burner before doing this step, as alcohol is inflammable. Now, what will you notice? The color of alcohol becomes green, whereas the leaf itself loses its color and becomes decolorized. Let us go to the next step, and the next step is that the leaf, which is now brittle, we take it out and we dip it in warm water to make it soft and permeable to iodine. We are going to do the test for starch with iodine. Then we place the leaf in a petri dish or on a tile and add a few drops of iodine to the leaf and note the color change of iodine on the leaf. What will be the color? It will change. The iodine itself is brown. It will change to blue-black, showing that starch is present. This is a diagrammatic representation of this test. You can see that leaf is being boiled to kill the enzymes, or you can do, to stop the hydrolysis of starch into glucose. Then we take the leaf out and we put it in alcohol. The Bunsen burner is off. Then we take it out and dip it in water to make it soft. Then we put it on a tile and put a few drops of iodine and test for starch, and the iodine, it becomes blue-black. So starch is present. Now, as I told you, we have to demonstrate that sunlight is necessary for photosynthesis. In the beginning of all these experiments, you have to make sure that starch is not already there. So we de-starch a potted plant by placing it in dark place for at least 2 days. Then remove a leaf and test it for starch to make sure that starch is absent. Sandwich the leaf, which is still attached to the plant, between 2 pieces of black paper. Fasten the papers with clips and place the plant in strong sunlight. You have cut a pattern on the leaf, and this I'll show in the diagram. After a few hours, you remove the leaf and test it for starch, and you will see the results. Your conclusion is shown here in this diagram, that where the black paper was cut and the pattern was cut, there, because sunlight was allowed to reach the leaf, photosynthesis took place, and only that area will turn blue-black. The rest of the leaf will remain brownish. Now, this is another way of demonstrating this experiment. You cannot, instead of cutting a pattern, you can just attach a black paper on the top and bottom of the leaf and anchor it with clips. And after the experiment, when you remove it and test it for starch, this area will not turn blue-black. The rest of the leaf has turned blue-black. The next test is for carbon dioxide. Here we take 2 potted plants, cover the pots with polythene bags, secure them to the stem. This prevents carbon dioxide from escaping from the soil. Place 1 pot in the bell jar, and you place potassium hydroxide in a dish under the jar, and this absorbs the carbon dioxide in the jar. Put soda lime in the flask on top of the jar to absorb carbon dioxide from the air. This is the diagrammatic representation. We do not allow carbon dioxide to reach the green leaves. And in the test experiment, the control experiment, we do allow, and instead of putting potassium hydroxide in the Petri dish, we have put water. So we have blocked carbon dioxide in our test experiment, and when we test for starch in the leaf, after exposing these potted plants to sunlight for a few hours, we test for starch, and starch will not be present when carbon dioxide was blocked. It was blocked from air coming into the flask from atmosphere, air inside the flask, and air from the soil of the plant. Where in the second bell jar, carbon dioxide was allowed from the air in the flask. So it will show starch will be present. So these are the steps as I've explained to you. And what is your conclusion? That sunlight is essential for photosynthesis. Another way of doing this is you just attach a leaf which is still there on a potted plant. Half of it is dipping in a jar containing potassium hydroxide, which absorbs carbon dioxide. So when we test it for starch, the part which was not getting carbon dioxide will remain the same, and the part which was getting carbon dioxide from the air will turn blue-black. Now, the third thing which we have to test is chlorophyll. Chlorophyll is the green pigment. It cannot be removed from plant without killing the plant. Instead, we use variegated leaves. What are variegated leaves? They are shown here. They can be used, and they have pale parts which do not contain chlorophyll naturally. The green parts contain the chlorophyll, and they will serve as the control. Now, which area? We do the same test, like we first test for starch, and then which area will be turned blue-black. You can see in the second leaf, those areas which were green in the first part have turned blue-black, which shows that chlorophyll is essential for photosynthesis. So some examples of variegated leaves are there during the plant, and there are some plants which naturally have variegated leaves. So these were the series of tests, and these are the steps here. First, you destarch the variegated leaves during the plant, then expose to sunlight for a few hours, then remove, make a drawing of the leaf, kill the leaf or boil it, decolorize it, and test it for starch. Later on, you make a drawing of the leaf to show the distribution of the areas which turn blue-black. This is another diagrammatic representation of the same experiment. And this is another kind of leaf which is variegated. And you can see those areas which were yellow. They did not have chlorophyll, so they are not turning blue-black. So this is all for today's lecture. This chapter has many other experiments, and also we have to describe the role of the leaf with its parts. I'd like to teach you all those parts on Valeem.com. Here is the information where you can approach me. Thank you for your valuable time, and I hope I'll see you in future. Thank you.